think long read is going to replace any kind of short read technology for any application.
It's just about time,
My bet is in 5 years' time there won't be any short read tech
right?
in the market.
So, Mr. Kumar, welcome to our podcast. The idea is to talk to founders from the biotech space, to founders in the biotech space. Think about it like that. And the goal is to really understand all the stories you have to tell from your personal journey and things that people could actually learn from all of it, right? So this is it. There is no starting take or there's no action point. We've been talking for a while, so we'll just jump right into it. I have like a bunch of questions, but before we get into any of those, I would love to hear from you, your version of your story. Why did you start what you started, right? Talk about Buck Speaks, talk about Leucine Rich. Tell us a little bit about the background and what motivated you to get into this.
First of all, thank you for inviting me. To this podcast. I mean, honored to be here. Wanted to do something in drug discovery. So my two co-founders, Prabhat, Dev and I, we were in drug discovery throughout our working careers and wanted to do something in preventive healthcare. Thought a lot about genetics. Then back in 2013,
2030, right?
there was this new thing called as microbiome,
Mhm.
which I thought was pseudoscience in the beginning. I was like, all of this is humbug. I mean,
Look
this
where
is,
we are.
uh, and thinking, can we use these tools to do preventive healthcare? So actually, it was me who took the decision, saying that let's do genetics, let's not go after microbiome.
Hmm.
So for the first couple of months, we were actually looking into genetics a little more. And then, as we looked more and more into it, kind of realized that, I can tell you you're a good metabolizer of fat or carbs or anything
Right.
like that. But there's nothing more that we can do post that. I mean, back then, even editing of genes
Hmm. It
was
was,
not there. I mean, it
yeah, it
wasn't
was
prevalent. So,
sci-fi. That's it.
yeah, it was sci-fi.
Nothing
It
more than
was,
sci-fi.
yeah. So, and then started looking at the microbiome and thought, this is a good tool to do something preventive. Maybe it's pseudoscience, but let's see if the science evolves. And that's how we got into this. And now I'm a microbiome evangelist. I mean, I believe in it so much.
Right, right. And how, like, what everybody has a story, that initial spark or that decision making story, right? Suddenly one day you realize, but it usually doesn't happen like you wake up with an idea. Certain things lead to it. So what were the triggers that actually pushed you out of your very successful career at that point to get out and do your own thing?
I was in drug discovery all my life. I mean,
Hmm.
waiting for the disease to come in and then say,
Hmm.
so a doctor to diagnose it, and then after that come up with a pill and
Right.
to cure it. Was sick and tired of those stories. So wanted to see if I could catch the disease very early, can prevent it, can reverse it.
Hmm.
Those were the thoughts that were running in our head to start the company. So the initial idea was to build a SaaS platform. Anybody
Okay.
would upload
Their data
any kind
or
of
their
yeah
genetic data, right?
yeah, any kind of NGS data, any kind of sequencing data will make meaning out of it. So that was the thought process, almost like that.
And was 23andMe already around? Right? It was around
It
in
was around,
2013. They were trying to do a little bit of that, right?
yeah, but our platform was more targeted toward researchers.
Hmm.
We were like,
Okay.
you want deeper meaning into the data that you're generating. I
Right.
mean, that's what we want to do.
Because you already come from that background
Mm.
where you analyze data for drug discovery
Yeah,
primarily. Got it. And around 2014 is when you committed, you
yeah.
said, right?
So Prabhat and I said, let's start a company. Let's officially register for it.
And your first idea was, like you said, to analyze incoming data
Yeah.
primarily for B2B like scenario,
Yeah,
is what I'm
so
guessing.
it would be researchers in academia,
Hmm.
researchers in universities, in biotech, small biotech companies. All of them, they would just upload their data, we'd
Mhm.
analyze it, send it back to them, and that's when we started getting a lot of microbiome data as well.
Okay, so that
And
happened to be uh, a side effect of what you were doing.
it was we were focused on human health,
Hmm.
and we wanted to do genomics, RNA-seq,
Hmm.
microbiome related to human health. We wanted to create a product, but as we were getting more and more microbiome data, all of us began to understand
Mm.
what was actually happening. And quickly, very very quickly, I learned it wasn't pseudoscience. I mean, there was
Mm.
something very very meaningful that is happening. Fast forward to 2024, we still, all the world's knowledge put together, we still understand less than 10% of the gut microbiome.
Okay.
So imagine back in 2014, 15, how
Right.
little people understood, and any little information that I could provide as a service
Right.
was very very meaningful.
And at what point did you decide to go to, I mean, whatever you're doing today, right? Like going all the way to the end customer, which is a patient.
So one thing about the SaaS platform, I mean, even if you just take the microbiome per se, was that every project was very unique.
Mm.
So there was a researcher working on cancer wanting to look into the gut microbiome. There was
Mm.
another researcher looking probably at the oral cavity and looking at the oral
Right.
microbiome, kind of thing. We even got weird projects like the spider's gut microbiome to see why the spider is eating a particular kind of bee in a farm,
Okay.
which is producing a lot of honey.
Right.
So the
Oh wow. Okay. That's a long correlation. Okay.
correct. So the amount
Right.
of data we had to crunch for that
Mm.
and try and make meaning out of it was crazy. So everything, it wasn't a platform; it was more a consultancy
Mm-hmm.
job, and that's where I knew that the scale won't happen in this.
Mm.
There's
In
no
in that mode of
in that mode of
operation, right?
things. So and at the same time, we got connected to a few people in Bangalore who were looking into who are training athletes
Mm.
um, who represent India at the international level.
Okay.
So these are
And they're like coaches or like
so they
personal
yeah so
trainers, right?
they're like personal trainers and more from a health point of view than technical
Mm.
trainers. I mean,
Right.
so they were asking us. You all know so much about the microbiome. Can you
Mm-hmm.
all test the athletes and
Mm.
tell them what they can do to improve their
Right.
performance? Um, and
That's a
that's
big
when we,
question.
and it's like, okay, yes, I mean, it's possible, but how do we collect a sample? How do we
Right.
do the sequencing? So
Right.
that's when we started stitching everything together and putting out a
Right.
test.
Now, fast forward to today. So, how do you operate? How many customers do you have, or at what scale do you test? Maybe you could tell a little bit about that.
We have over 200 partners from India, Thailand,
Mm.
Philippines, and some other Asian countries as well. So these partners could be, big hospital chains
Mm-hmm.
with having maybe 30, 40 centers around India, those
Mm-hmm.
kind of hospital chains, to a single nutritionist probably getting about three or
Mm.
four clients a month. So
Right.
right now, doing close to about 4,000 samples a month.
Wow.
Um,
That's a big number for sequencing.
that's a big number for
Any
sequencing. I mean,
kind of sequencing.
till I mean, this jump has happened over the last 6 months. I mean, last year it was us doing 40 samples a month itself was a big
Mm.
deal,
Mm.
and so from there to jump over here was is
That's
all
great.
it's an effort. It's the fruit of the effort that we've put for all these
A
years
few
to
years, yeah,
to get
definitely,
here.
right? All through COVID, before that.
Yeah,
I think you were talking about sequencing genomics 2014. Just imagine how many, it feels like decades passed because of COVID.
yeah,
And suddenly, then COVID hit, and the knowledge, or at least the general availability of knowledge around sequencing, I think scaled up really well around COVID, post COVID, just like how PCR tech became
right.
ubiquitous only after COVID, right? So we've noticed that. I think maybe India had a few hundred PCR machines before, but now there are few thousand, and that's a big jump. And suddenly, everybody wants to do a molecular test.
Right.
If you can do it in a molecular way, do it in a molecular way. And I think similar stuff happened to sequencing also. And I think it's for you also to take in all that timing and your effort, collective effort of years, and now you're suddenly at this high load. I can imagine that race would have been
So
crazy.
imagine before 2019, before 2020, actually, we were collecting poop samples
Hmm.
and shipping it to US for
Oh
sequencing.
wow! So you get
Um,
the data back,
so we get the data back. I mean, we couldn't get a sequencing center in India to do it for us at the cost that we wanted to.
right?
So that's how tough it was. And so I've sent it to the US, I've sent it to Singapore, I've sent it to Hong Kong.
Hmm.
Um, We had special relationships with FedEx. I mean, the only guys calling up saying that, I want to export shit and
Yeah. From India
from
out.
India. So how do we do?
Literally shit.
So that's been a journey, and
Right.
and 20 late 2019 is when we realized that we've hit upon something, and we wanted quality under our own control. So
Hmm.
till then, we were purely a computational biology
Right.
lab, uh,
Primarily a software company in a way.
primarily a software company. We said, let's build our own lab. I mean, how
Right.
difficult can it be? So we bootstrapped our own first
Hmm.
sequencing lab.
Hmm.
Um, we went into the first principles of almost everything.
Right.
Why is a vortex needed?
Right.
Why can't you just
Yeah.
spin
Yeah.
it with your hand?
Yeah.
So
Yeah.
we actually got people passing by the office to spin the tubes for us,
To
and
test the difference between with and without
with
vortex,
and
right?
without.
Yeah.
So we did that for all the chemicals. It was, I mean, we tried everything. We questioned everything, and we started our own lab. uh, which is why for me, unit economics
Yeah.
was the biggest thing.
Absolutely. No doubt, right?
And we worked at it, and that's how we've come this far.
Right. And now, do you run most of your sequencing in house or do you still outsource a small part of it?
100% is in house,
100% is in house.
unless it's coming from a country where I can't get the sample out of the country.
Okay.
For
And you get it
example,
done somewhere and
yeah,
get the data.
for example, Dubai, they've over the last year
Oh, really?
they've stopped any kind of biological samples coming out of. The rule is any Emirati sample should not go out of the
Okay.
country.
If it's an Emirati sample,
Yeah, but it's difficult to prove that, right? I mean,
you might as well just stop everything. So,
stop
yeah,
everything. So
makes sense.
there we've partnered with a local lab over there to do the
Okay.
sequencing. So otherwise, we try and get back to Bangalore.
Awesome. And so, do you use long read sequencing or short read? What's the.
In 2020, we did close to about, I would say close to about 1,000 samples, anywhere between 700 to 1,000 samples. We tested both on the short read technology
Okay.
and the long read
All of
technology,
those samples.
all of those
Okay.
samples parallelly. And we found that long read gave us much better results than the short
That's
read.
that's a big thing to say, right?
Yeah.
Like a lot of people wouldn't have spent so much to actually come to that conclusion. It's easier to just assume something and run with it because everyone else is doing.
Correct. Every salesperson has their own
Hmm.
point of view and their own
Right.
pressures, and
Right.
we actually wanted to see the data, compare it a
Yeah, you
little bit,
wanted
and
to
then
dig the truth out of
uh,
that scenario, right?
and see what can be done, and that's how long read was preferred, and it's I mean, it was a good decision on our part. I mean, at that point of time, we were breaking our heads a lot
Hmm.
on where to go with it, but once
Which direction to
which
pick?
direction to go, because it's a commitment, right? I
It's a
mean,
very big
I
commitment.
mean,
Once you're in it, you're stuck. Yeah,
correct. Till we were only a software company. I mean,
it was
I just had to
easy.
change the vendor, and
Yeah, yeah.
and it got done. But that was a big commitment. So we wanted enough data to actually say that this is the direction that we were heading, and uh, it's proven to be good. And now I think long read is going to replace any kind of short read technology for any application. Um,
It's just about time.
my bet is in 5 years' time there won't be any short read tech
Right.
in the
Yeah.
market.
And I think even Illumina is coming out with a
Yeah.
long read platform. MGI actually beat Illumina with a long read platform. It's crazy. I can see this moving around everywhere. You're
Wait till
so right.
Roche releases its long read
Roche?
tech. Yeah, it's
Roche has purchased a few companies and it's been at it for a while. And do you think there'll be a scenario where sequencing test, something that you do, um, barring the software analysis part of it, do you think it can happen on site?
Oh yeah, yeah, but definitely. As in, uh, look at it outside the healthcare realm now.
Hmm.
A lot of the agriculture is happening on site.
Really? Hmm.
At least small time agriculture, all of that. I know a company in Ooty right now, uh, which I sorry can't name, but they're doing a lot of the sequencing at the compost site,
Wow.
and and companies like Oxford Nanopore is enabling that, right? I mean, they're saying sequence wherever you want to sequence,
Mm.
kind of thing. There's a lot of tech that is now going into.
And what exactly are they looking for when you say
I mean,
compost site?
uh, they're looking at microbial populations,
Right.
and they're looking at how to improve it, how to improve agriculture based on that. Look at how NASA's taken
Right.
the nanopore to the International Space
Right.
Center. So this I think on site is going to happen very very quickly.
It's exciting, right? Then most of the work will also be on software,
Yeah,
which you've been at for the longest time.
correct,
Sequencing in real time, analyzing in real time, turning that into an actual report that someone can sign off on. It's a big space.
correct,
I think there is one company called DNA Nexus
correct.
which does phenomenally well. I still struggle to understand. I mean, it's a great success. They have 100 million in revenues just selling software for sequencing analysis,
Mhm.
which is something that you've done yourself, right? And we live in a world where TCS, Infosys, all these guys exist and do so well in India. Um, why do you think such software companies have not come up yet?
Because what DNA Nexus and a couple of other companies in this, I think Genius and a couple of other companies that have done well in this area is that they've combined the hardware with the software.
Mm,
The one thing that researchers,
mm,
cloud is something that they're scared of. I mean, data privacy, data infringement,
mm,
those kind of problems are there. Secondly, NGS required at least till recently it required a lot of RAM to do it. I've done
mm.
projects where I had to use a 1 terabyte RAM on AWS, and it ran for 15 days. And I
I can
and
imagine
I
the cost.
undercoated that project, and we lost money on that. But it was a
Wow.
learning.
Right.
15 days, 1 terabyte RAM on AWS. But that was the RAM requirement at that point of time. Now, with we also dabbled in a little bit of FPGA programming
Mm,
and all of that. And with that South Korean company, Illumina bought a Dragon.
mm.
That processing has come down
Right.
significantly. There are other improvements. GPU,
Then the
um,
GPU era
yeah,
now that we live in, right?
GPU era came into genomics in 2017 itself.
Mm,
The general purpose GPU programming, CUDA
right.
programming was there for the longest time in bioinformatics. I think all of that has now become better. So back then, I don't think software companies thought of the hardware problem
Correct.
that was there.
It's just like any other industry that
Correct.
way, right?
So if I remember reading an article saying that the Craig Venter Institute still FedEx hard disk across institutes. This is again back 2017, 18,
Right.
rather than doing it on the
Yeah,
internet.
it's probably faster.
Yeah, genomics is going to be the second largest data set after the astronomical data. So
Yeah, that
imagine.
suddenly shifts the perspective, right?
Yeah, so I think there's a combination of hardware and software
Mm,
that was needed, which wasn't provided. Now I feel we're slowly getting into the era where there's going to be a lot of specialized software,
mm,
like ours
of course.
and many other, which is going to do analysis. But I think the struggle is going to be in the signal outputs that are going to come from the machines, because the machines are going to be so varied. The signal outputs are going to be so different. I
Right.
think that's where the opportunity is right now.
Yeah, whoever captures that width
Correct.
soonest, right? Which is not easy, right?
Yeah.
If you have
It,
to run a validation on Illumina versus Elemental versus Oxford, all within a time span, you literally have to multiply your teams into those many platforms.
but you're still talking about secondary and tertiary analysis.
Right.
I'm still talking about primary analysis, which is
Directly from the machine itself.
directly from the machine. There's so much of data that is getting missed out. Even with the ONT's case,
Mm,
it actually the FASTFI files actually have a lot of data in it.
mm,
What we extract is very little out of it,
right.
which eventually becomes the FASTQ. So there's a lot of scope. There's a lot of machine learning
Mm.
that can go into it, and that is an area where specialized software can actually come into play.
There's this whole idea of trying to get the higher fragment sizes out,
Hmm.
right? Like eliminate as much as lower fragment out. We spoke about this maybe a year ago. Um, You can maybe tell us a little bit about why it's important. Why does it matter?
Okay, it's important for all samples
Right.
except for stool samples.
Oh, is it? Okay.
In fecal samples, you have no idea what's there. I
Mm.
mean, now we've seen so much of poop that as soon as I look at the sample, I'll tell you whether the DNA quality is going to be good or bad just by looking
Okay.
into the
Just how
sample.
it's stored and how it's come. Yeah.
Yeah, just
Yeah.
just by looking at that. Let me talk about other samples first. I
Mm.
mean, there from a bioinformatics point of view, and from an accuracy point of view, the longer the DNA length, the more accurate the alignment and the readouts that come out of it. So purely from that point of view, I would say go for the
Mm.
higher length as much as possible. Again, there is also a challenge of being too high. Sequencing errors do
Mm.
tend to come in where the read length is very very large. I'm talking about 1 lakh bases and above
Okay.
kind of thing. Um, in fecal samples, the challenge has always been trying to. So we can't remove the low fragments.
Right. You
For
can't
us,
remove anything.
I
It's
can't. I can't.
risky to remove anything there.
Correct,
Yeah.
correct. Risky to remove anything. We try and capture everything that is possible, but we try and keep the N50. We try
Mm.
and increase the N50 as much as possible. So
Mm.
from a microbiome sequencing point of view, there is not too much of innovation that we're doing in the sequencing, the library prep and sequencing per se, but we're doing a lot more on DNA extraction,
Mm.
and that's
Correct.
where the biggest challenge is.
Right. It still seems, for anyone looking from outside, seems like extraction is such an old thing. Like, what more can you do in it? But as a company which spent, at least us, the last 3 years pretty much only obsessing about extraction from different sample types, we understand. And I'm happy to hear it back from you. It's like validation of all our efforts, right?
Yeah.
It can
So
make or break the rest of the process.
correct. I mean, so you change anything in the chemicals, you might bias over gram positive versus gram negative bacteria.
That changes your report altogether.
Correct. You solve for that, then there might be a bias between bacteria and fungus.
Hmm.
So trying.
And there's a risk of not even getting fungus because you've not lysed
Correct,
properly. So,
correct. And short fragments again, you only go for large fragments, then you're missing viruses.
mm,
Because most of the
absolutely,
virus fragments
very
are
small.
very small. So, but, with all of this in mind, and then you take for us, you take the fecal sample into consideration, then your it goes exponentially high. All the
Right.
problems that come with it.
And it also can't take too long
I
as a
can't.
process. Right.
It can't take too long as a process. Can't be too cumbersome. You have too many pipetting steps. Then there's biases that are going to come from that. So all those kind of things put together,
Right.
and it's still unsolved. I don't think we still keep
Mm.
experimenting to make sure that
It can be improved.
how
There's
can
still
we improve.
room. Right.
There's still room to be
Absolutely.
improved.
How you spoke about ultra human just some time back. They made the whole tracking glucose cool, right?
Hmm.
They made it a athletic element. They added that whole sportsman thing to the idea of tracking your glucose. I when it started, they never told anybody that it's a diabetes management
Mm.
company. That's something that they've done very differently. They just put themselves away from the diabetes management crew and they said, you know what? Anybody young, passionate about health, you want to hack into your system, start with tracking your glucose. Microbiome is also like that
Mm.
in a lot more ways, right? In fact, you said your idea actually came from trainers coaching
Correct.
athletes. That's kind of how it started. Now, there is another extent to it. Microbiome directly helps depression, directly attacks your overall immunity. There are many ways it directly impacts. Have you seen in your personal journey, like, have you seen any patient or an end user or a system where this data directly impacted a health condition?
Oh yeah, yeah, tons of it.
Is
As
there
a,
any
so
one story that you would love to talk about?
a paper on diabetes and all that. There was a high flying CEO. She was traveling different countries, um, very health conscious,
Mm.
ate 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 the right kind of food, um, but because of her work, the work pressure, all of that. She thought it was because of all of this that her health was getting messed up
Mm.
all the time. And we got her sample. We went through the data, and the data was showing it to be very, very different. It had no stress markers in it. It had there was nothing about stress or anxiety or any of those kind of things. What we realized was her diet was she was so health conscious
It
that
was perfect.
it was perfect. I mean, it was the same 4 5
Right.
items over and over again. So there was
Right.
absolutely no diversity in the
Mm.
gut, and that was causing more problems than anything else. And there was absolutely no pathogens in the gut, which is also alarming.
Mm.
So in my world, there is no good bacteria, bad bacteria.
Right. Right.
Everything is in relative proportions.
Right.
You need to have trace amount of
Right.
E. coli also in
Right.
your gut. So apart from actually advising her to eat a lot of diverse food, don't binge only on broccoli.
Right.
Please,
Right.
eat many more vegetables, fruits, cereals, and all of that. So that was one advice. The second thing, which was the harder thing for her to do, is like every once in a while, maybe once in 3 months or something like that, go have a roadside pani puri, please. Uh,
So this is like your favorite recommendation. Right.
um, but you have to be careful who
Right.
does it
Of course.
and when they do it and
Yeah.
all of
Yeah.
that. There's a very, very thin line between infectious disease
Mm.
versus having good diversity.
Wow.
So how do you stay away from infectious disease but have a good diverse microbiome is the key.
Wow.
And
That's a great point. I'm really looking back at what I ate in the last 1 week, and
think about it. All of us tend to think that we eat a lot of vegetables. Think to eat that
we
we
don't.
eat the greens and all that. But it's the same 5 6 vegetables that we are
Yeah. Yeah.
repeating over and over again.
Yeah.
Sometimes I throw a challenge saying have 30 vegetables
In one day, or like
in
in
a
a
month
meal.
or something like
Right.
that
Okay.
in a certain period. I can't even name 30 vegetables. So
Yeah. Yeah. I'm
uh,
pretty sure. I can't.
uh,
There's no way I can. Yeah.
we've lost seasonality in our
Mm.
food. We've
Right.
lost a lot of that. So we have to be a little more conscious into getting diverse food items into
And
in.
when you look at these samples and data that keep coming through your door, right? Are most of your recommendations about imbalance versus balance? Is that generally what it is? Or can you actually point out and say that this particular area you're super deficient, you'll have to go up here? So, how is it like?
So both of that. So one is looking at the larger picture of imbalance versus and trying to balance that out.
Mhm.
And so the the algorithm that we have built is we know which food will starve a bacteria and which food will
Okay.
grow certain bacteria or
Okay.
fungus. So based on that, we give personalized diets.
So you actually tell people maybe eat more of this and not more of that.
Yeah,
Okay.
stuff like that. But then we're also looking at specific things,
Mm.
saying that maybe this pathogen is overgrown, and
Mm.
hence you're having this problem. So these are some of the natural antibiotics that you can take, or these are other bacteria that can go into your system that will fight this
Mm.
pathogen. Or in certain worst cases, this person actually needs a very targeted antibiotic
Oh
that needs
wow.
to be used.
That's so high that you actually need an antibiotic.
That you actually so then we recommend a gastroenterologist will probably look into
Okay.
the data and prescribe the right kind of antibiotics over there.
And in
But
that
those
case,
are
the report goes to the
yes,
gastroenterologist. Okay.
yes. So which is why the report is so verbose and so lengthy, and
Mm.
because there's no one size fits all kind of
Right.
an answer for this. Everybody's the nutritionist is looking at it from a slightly different angle.
Mm.
The doctor's looking at it from a slightly different angle, and you need all these inputs to make your health, to make your gut better.
Right.
Um,
And this is coming back to your partners, right? You said you mentioned you had 200 partners who actually bring in samples to you on a monthly basis. What percentage of it could be a hospital, and what percentage could be like a nutritionist? Because in my mind, these two are very different sources.
so till about early this year, it was 70% from the wellness community.
Mm. This
So
is
when
nutrition, sports,
sports, that kind
general
of
health and wellness.
when nutrition, when diet is the most important thing,
Mm.
and maybe 20-30% was the clinical
Mm.
community. But as we are publishing more and more papers and proving what we're doing, the clinical community is opening up a lot more.
Mm.
I think by in about 6 months' time, it'll be 50-50. It'll be
Oh wow.
the clinical community doing 50 and the wellness doing 50
But
as
I
well.
think when clinical scales, it really goes up, right?
It really goes
Then
up.
it becomes like a some doctors recommend it automatically. More doctors will recommend it, and
Correct.
it becomes a prescription after a point.
Yeah. Right now, what's happening is a lot of the patients are actually going with the gut microbiome report to the doctor saying
I
that
already have
I
this.
already have this. Can you make some recommendations? So once the doctor sees about 5 patients coming in
Mm.
with the report, and then he also sees he or
The
she also sees
response.
the response and how it's actually impacting the disease.
Mm.
And that's how they reach out to us and they say, we want to utilize your test.
That's great, right? Like someone actually calling you and saying that I've seen your report, I've seen it two, three times. Tell me how can I actually prescribe it?
Great.
Yeah.
And they're also adding the point that it's actually helping my patients.
Mm. They're
Um,
happy to
yeah.
bring
So
it in.
unlike on any other diagnostic report, we can't give a yes or a no answer in a
Correct.
microbiome report. So, which is what the doctors would ideally want, right? Yeah,
Actually, yeah. They
given
let them say yes or no or take the decisions.
yeah, given their time, their work pressure, and all of that, they would prefer a more simplified answer.
Mm.
Unfortunately, the microbiome is so complex that you can't give a very simplified answer. You'll have to take two or three things nuanced together and see, and then take a call
Mm.
with other medical history.
Right.
So that's something that they're now willing to accept. Realize some doctors that we work with have they're putting together an entire team
Oh
which
wow.
looks into the microbiome nutrition,
Mm.
um, as well as doing their normal practices as well.
And I'm sure there are these teams approaching care
Mhm.
generally, especially in case of say oncology, cancer. I'm sure there are teams out there. Again, it's an assumption I'm making out loud who actually work with oncologists and microbiome data and try to help the patient through their journey. Have you seen anything like that?
We've started. I think there are 3 studies that is ongoing right now, which is on oncology
Hmm.
related. Before the cancer, I mean, when the cancer is developing, after the cancer is developed, during therapy, after chemotherapy, after all of that, the microbiome plays a huge role. But it's very, very difficult to directly go and put
Hmm.
the microbiome test and therapy in in the patient
Right,
population. You need to do a few studies. You need to understand what needs to be done the right way,
right.
and those are the studies that we're doing right now.
And
Uh,
then this is what you keep publishing
yes,
year on year.
yeah.
Okay.
Well, I believe in it. We as a team believe in it. We're getting our NABL and NABH for the first time in India
For the lab, right,
for the gut microbiome test.
right,
There were ways to circumvent and
right,
and get the mark, but we
right,
wanted it for the gut microbiome test.
right.
So we've been educating the regulatory authorities who have come saying that this is what we do, this is the protocol that we follow, these are the standards that we think needs to be set.
Hmm. So how hard has it been regulatory as a challenge?
The problem with the microbiome, I mean, not only in India, worldwide, it's been in a gray zone. See what benchmarks can be put over there, and then my prediction is within 2 to 3 years time, you will see a lot of FDA approved, CE approved
Hmm.
microbiome solutions coming up.
But coming back to TGA that you mentioned, right? Australia is one of the very few countries which actually sponsors a colon cancer test.
Mhm.
After I think 55, or there's an age limit after which everybody in the country gets a free colon cancer check. You basically send your stool sample, it gets collected at home, gets shipped back to a lab, and then you get a report and you just do a consult. It You can always say, hey, there are not many people. It's easy for them to do it, but we should also take time and appreciate the forward thinking over there, right? They're actually believing in a genomic test, in a predictive scenario, and actually taking action and finding more and more evidence year on year. Do you think India will get there, or are there people working in that direction with insurance or government in the genomic space?
I'll get to that question in a bit. One interesting tidbit about Australia with respect to the colon
Colon
test.
cancer, yeah.
So, I was talking. to one person from Microba
Hmm.
a few years ago, and I was asking them, how did you all manage to scale so much in Australia in such a short time?
Hmm.
Microba, I think, was one of the first microbiome companies worldwide which went public, right?
Right.
They did their IPOs and went public. So I said, how did you scale? What's the secret sauce? How did you get it? And there were many answers in terms of technology and how they went about market and all that. But one interesting point that came out was people are used to giving stool samples. At least the senior population are used to giving stool
Because of
samples to men.
this colon cancer
Correct.
test.
Here, we're trying to make collection of poop samples cool.
Hmm.
We're trying to
Hmm.
make the first kit box we had was a wooden jewelry box,
Oh,
saying that your
really?
poop is so important,
Right.
for it. So that was one of the things
Hmm.
in Australia. I don't know. In India, it's a little difficult. I mean, because ICMR, CDSCO, they're also playing catch up. Uh, there's so much work that they need to do. So I don't know whether regulations will be forward thinking in that way. They might be uh,
Dependent on
dependent on
someone
other
else.
agencies coming and giving them recommendations.
Right.
I'm not sure. I don't track the Indian space
Hmm.
as much as I would like to, but it'll be nice if we can take the leadership in it. If India can take the leadership in it in some of these things, but it's a big ask. I mean, the
Hmm.
government has so many other. The public health itself is such a big thing in India that they have to do. I don't know
Yeah,
how
this
much.
size is so hard. The
Yeah,
volume is so hard to manage.
and the differences are so much
Hmm.
in India that it's a tough job for them to get this done.
What's the future of Leucine Rich, and where do you see yourself going? And what's the most exciting project that you're now, you have your eyes on today?
We want to keep expanding on the microbiome space. So the most exciting thing is, I think by the time the podcast comes out, we would have launched the skin microbiome test.
Oh, wow. Okay.
And when I say skin, I mean face
Hmm.
to begin with. So we've done a lot of studies on it, a lot of work on it. So
That's exciting.
want to now
That's
get
a
it into
that's a
the
dermat prescription almost at some
no,
point.
we want to go one level further and go to the consumer and tell them that
Right.
this is what you can learn
Right.
about the skin microbiome.
Awesome. So
Uh,
is it like a self collection?
yeah,
So they swab their
they
face.
swab the cheek and cheek face wherever, and
Okay.
then send it back, and we do the sequencing and.
Wow. So this is when we call actors and actresses, right?
But I mean, we've now developed a report which is 10x better than any other report that's out there in the world, and it's taken us 2 years.
Hmm.
We wanted this out much earlier, but it's taken us 2 years to really nail down every different aspect of the report and get it out there. We're doing a few projects on the inbuilt microbiome, so taking a swab of the room and seeing
Ah,
what the microbiome of the room is looking
right.
like. I know companies which are trying to put probiotics into ACs.
What? Wow. That's crazy.
I think it was Unilever that's come out with a
Mm.
probiotic cleaning surface cleaning product in Indonesia.
So you
Uh,
don't kill the good,
you don't
you
you
kill
don't
the good bacteria.
yeah, you don't kill the bad, you just apply the good or something like that
Oh,
is
wow.
the mantra. So we're doing few of the projects. I don't know whether it'll ultimately become a test that anybody can use, any consumer can use, but maybe, I don't know. Let's see
Yeah.
what it entails.
Definitely something to look forward to for enthusiasts. They
Yeah.
should keep looking at your website to see updates.
Yes.
Awesome. So I know that you're going to go back super active on LinkedIn very soon, and I hope this becomes one way to talk about all of your endeavors and then get into further details. It will be impossible to stop the podcast. I kind of gave you a warning, but now with the time limit, we should probably call an end to this particular episode. But I'm super excited to hear more about things beyond this, and I'm sure there will be another follow on episode to this after you launch the skin microbiome. That's something that's super exciting. Thank you so much. Thank you for taking this time, coming all the way. I know video is hard. You did tell me it's a lot more stressful, but this is the beginning of a new thing, I guess. Any parting words for the audience?
You know, Thank you for inviting me. I think our partnership with Cambrian is only going to grow. There are so many synergies that we have, and this is what I think the biotech of India needs to do: collaborate, build a stack. I mean, the tech world has a tech stack.
Mm,
Let's build the biotech stack. Complete offerings that we can give to the rest of the world is what is needed. Um, Bangalore's a cool place to do
mm,
it with so many startups having so many innovative ideas.
right.